Hello, everybody. Welcome back. This is our last installment of our lessons for Chapter 11, the Genetics and Inheritance chapter. I will have a few more lessons from Chapter 16. We're only doing part of that chapter, so we're not completely done with genetics. We have a few topics in Chapter 16 focusing on human inheritance for that one, so stay tuned. So in this last piece, we're going to, again, extend away from Mendelian inheritance pattern with the truly dominant recessive, homozygous, heterozygous kind of foundational ideas. We added some complexities with linked traits or linked genes and X-linked genes. This one, we're going to just take a look at some variety that, again, Mendel luckily didn't have, the P plants didn't show any of this. Otherwise, if it did, he may not have been able to come to his conclusion. So these complex inheritance patterns have been identified elsewhere, and we're going to just take a look at some examples. All right, so the first one is taking a look at X chromosomes. So in this picture, first of all, this is not clearly showing all of our human chromosomes. It's just showing the X chromosomes in a female. This would not happen in males. This is only a thing that happens in females. Because females have two X chromosomes, just for the X chromosomes, it's not true for any of the other autosomes, the 1 through 22 chromosomes, is that one of the X chromosomes gets turned off. It's called X inactivation. And so this randomly happens very early on in embryonic development. So it's a random turning off or inactivation of one of the X chromosomes. So for example, we have a maternal and a paternal, so the pink and the blue. So one possible outcome is the pink chromosome gets inactivated and the blue chromosome is what's left to be expressed. Or you could get the blue chromosome is inactivated and the pink one is left to be expressed. So again, this is very early on in embryonic development, like early cell stage, like maybe the 4, 8, 16 cell stage. So once those X chromosomes are inactivated, they stay inactivated for the remainder of the life of that particular cell. So whatever daughter cells those cells make, that X chromosome will be inactivated for the entire cell line. So all of the ones that have the blue cell inactive or the blue chromosome will be inactivated. All of the daughter cells of that cell line, the blue chromosome will be inactivated. For the ones that had the pink inactivated, all of the daughter cells will have the blue expressed and the pink will be inactivated. So these are going to be making all of the cells in your body. Now, normally, if you have the different alleles, right? So if you are homozygous recessive or homozygous dominant for both of those, it's not going to be a big deal because you will have the same allele on the active ones, no matter whether you're the pink or the blue. But the issue comes, or the observations, I guess, come if you are heterozygous. So let's say on this chromosome, we are capital H for something. And on this chromosome, we are lowercase h for something. So the parent cell was heterozygous, diploid heterozygous. Now, all of these cells are going to express the dominant phenotype, and all of these cells are going to express the recessive phenotype. And all of the cells, no matter where they end up in the body, that's the allele that's active in those particular cells on the X chromosome. And again, it's just dealing with the genes on the X chromosome. So I have a couple of examples here. I have the kitty pictures. Calico cats are a result of this X inactivation in their fur color. So fur color genes are found on the X chromosome. So you might have an allele for orange color and you might have an allele for black color, and wherever those color pigment cells are located to give the fur its color, maybe this could be orange and this could be black. I don't know if that's the case, but let's just say. So all of the cells that got the orange dominant allele, wherever those end up on the body, they're going to make orange fur. And all of the other ones that have the black allele, wherever they end up on the body, they're going to have the black fur. So you get this combination of different colored fur around on the different locations of the cat. In a human example, it's more of a disease kind of situation is called anhydrotic ectodermal dysplasia. And this is a big scientific name to say malfunctioning sweat glands. So the distribution of sweat glands, the instructions for that are located on the X chromosome. Now, if they were heterozygous, so they have a normal functioning allele and this mutated allele, you might get normal sweat glands on any of the cells that are carrying the dominant allele, and you might get missing sweat glands in any of the cells that have the recessive allele. So what that means is these little patches are going to be made from the cells that had the normal X chromosome inactivated, and all that was left was the recessive. And everywhere there's normal sweat glands, everywhere else on her body, that's all going to be the dominant allele. So it's a really interesting distribution of normal or dominant and recessive alleles because of this X inactivation. And I have a picture of an actual, so these are just skin cells. These are like cheek cells. You might saw that in the lab. But right here, let's see, right here, that little dot right there, that is a bar body. So within the nucleus of this particular cell, you can actually see that tiny little black speck. That is the inactivated X chromosome that's just kind of squished over on the side. And in the rest of the nucleus, you just have that one X chromosome that's activated, and that's what's going to be expressed, and that's what's going to determine the phenotype for any of those genes that are located on the X chromosome. So that's kind of interesting. There's no way Mendel would have figured that out. All right, the next characteristic or complex inheritance pattern is called incomplete dominance. Now, this is where you don't really have a dominant and recessive. So I don't want to say homozygous dominant and homozygous recessive because there's no dominance and there's no recessive. And what you end up with is you get homozygous red, we can say, and homozygous white. Here again, we're just showing the superscripts to show the differences because there's really no capital or lowercase. And so you have homozygous red. If you mix that with a homozygous white, you'll still get a heterozygous, right? The 1, 2, you have one of each. But the color is not going to be either red or white. It's going to be an intermediate phenotype. So if you were to mix, right? So here's our parental true breeding parents. We get our hybrid F1. You cross the hybrids, and instead of getting a 3-to-1 ratio like we saw with the monohybrid cross, you end up getting a 1-to-2-to-1 ratio. So 25% of your offspring will be homozygous red, 25% of your offspring will be homozygous white, and 50% of your offspring will be heterozygous. And the phenotype is an intermediate phenotype. So your biggest clue for identifying incomplete dominance is an intermediate phenotype. So some examples might be straight hair and curly hair making a wavy haired offspring. Wavy is neither curly or straight, it's in between. That's a good clue you're looking at incomplete dominance. If you have black and white and you get gray somewhere in the middle as a phenotype, that would be a clue you're looking at incomplete dominance, or a 1-to-2-to-1 ratio. Okay. The next one is codominance and multiple alleles. So human blood type is a good example for both of these inheritance patterns. We're going to take a look at codominance first. Codominance means you have two alleles and one is not going to overshadow or be shadowed by another one. So in this case, our blood types, A, B, and O blood type. So A is dominant and B is dominant. So if you were to have A genotype, you would be blood type A. You would show the A surface proteins on your red blood cell. If you happen to have the B allele, you would be type B blood. But here's where the codominance happens. If you happen to be heterozygous for both, so this would be maybe an AA genotype, this could be a BB genotype, But if you are an AB genotype, you would have both A and B antigens. There's not one that's going to overshadow the other. So that's codominance. Both of the alleles are expressed in the phenotype. Now multiple alleles, now we have to add this guy over here. So for blood type, we have 3 alleles. 3. We have A, B, and O. But we only ever express 2 because we are diploid organisms. We're going to get 1 from mom and 1 from dad. Now we just have 3 choices. So up until this point, we've already just had 2 choices, purple and white, tall and short, taco tongue or no taco tongue, attached earlobes or no attached earlobes. It's always just been 2 choices. Well, not all genes have just 2 choices. There are some genes that have more than the 2. And so that's what we call multiple alleles. In a blood type, in our blood type example, we have 3 choices. We have A allele, B allele, and O allele. So we could have different combinations. And in this case, O happens to be recessive to both A and B. So what that means is we can now expand on the genotypes that would give us our various phenotypes. So while I have written homozygous A, homozygous B, and heterozygous AB for these blood types, we could also be type A blood if we were heterozygous for AO or BO. Okay, so an AO heterozygote will be type A blood. A BO heterozygote will be B blood. We can't have O in our AB because the 2 spots have already been taken up. But how do we get an O type blood? It has to be homozygous recessive. So little o, little o. I'm type O blood, so that means I got an O from my mom, an O from my dad. I have O blood. I don't have any A or B antigens anywhere in my genes. So codominance, because A and B are both expressed when part of the genotype. Multiple alleles, because we have 3 choices for that particular phenotype. The next inheritance pattern is called pleiotropy. So pleiotropy is one gene has multiple effects body-wide. So typically, again, up to this point, one gene, one effect. So one gene codes for earlobe or widow's peak or knuckle hair or hitchhiker's thumb or taco tongue or freckles or, all of the traits that you were looking at in your lab this week or some of the examples that I showed earlier. It was one gene, one phenotype, one effect. But that's not always the case. You might have one gene affects multiple parts of the body. The examples that I've given here, or the example that I give here, is a disease called cystic fibrosis. There's one gene codes for this particular enzyme or protein. It's called CFTR. Cystic fibrosis transmembrane conductance regulator protein. In normal individuals, it's normal. It's going to help, it helps pump and move ions across the membrane, like sodium and chlorine ions. Sorry, I had the knock at the door. Neighbor kids. Okay, so cystic fibrosis, the one gene codes for this particular protein. And if it's normal, your membrane transport mechanisms are normal. If it is mutated, which happens in the disease of cystic fibrosis, that mutated protein, which is found all over the body, has wide-reaching effects. So we can see that it affects your airways. You have this thick mucus that is produced. It affects your liver, your pancreas, your intestines, your reproductive tract, and your skin. So you have one gene that can have wide-ranging phenotypic effects. So one gene, multiple phenotypic outcomes if it is mutated or has some deficiency. That's pleiotropy. Okay. The next one is epistasis. So here's a dog example for all you dog people, right? I'm a cat person, but here's a dog example. So epistasis actually means upon. So epi means sitting upon. It's basically the effect of one gene or the expression of one gene can have an impact on another gene. So for one gene to be expressed, the other one has to be expressed in a certain way. So it's like a partnership. So in this particular case, we have different types of labs. We have the black lab, the yellow lab, and the chocolate lab. So we have the 2 genes that are kind of playing with themselves, playing off of each other. So we'll try to highlight those. So they're the B and the E. So here is our key, right? So big B or this. right? So that is the color part. So if we have, let's see, this is going to be black, and this is going to be brown, right? So the B codes for the color of the dog. We have black or brown options. You're like, wait, but there's a yellow dog. Where did the yellow dog come from? Well, we have this other gene. How can I write this? So this E gene, this allows for deposition of color. And this codes for no color, no matter what the color gene is. So for example, if we want to deposit color, so if we want to make sure that our dog's fur is going to have color, it has to be either homozygous dominant or heterozygous. So all of these guys that have the black or the brown fur will notice they have the big E, at least one big E. And then once that big E is found, we can deposit color, and then we look to the gene for the actual color to determine the color of the dog. So if it is, so I'll get rid of the E's. So if it is big B anywhere with a big E is going to give you a black lab. Little b, homozygous recessive, right? So homozygous recessive with a big E somewhere is going to give you the brown lab. Now let's take a look at the yellow lab. What's going on here? So it's yellow because if you look at the E gene, there's no color. It does not matter whether it's homozygous dominant or heterozygous for the brown or the black color, or even, brown or black. Homozygous dominant and heterozygous is black. Homozygous recessive is brown. It doesn't matter, no color. And that's how you get the yellow lab. So that's epistasis, because the expression of color is dependent on the expression of this E gene, whether the color is going to be deposited or not. That would be like in humans, like albinism. So if you have an albino gene, like if you're mutated in your albino gene, you do not produce pigment. No matter what your pigment genes are, you're not producing pigment. If you are normal in the albino gene, that's expressed, and then your color pigment based on your inheritance pattern will give you the color of your skin or hair or eyes. All right, so that's called epistasis. The next one is called polygenic inheritance. Now again, up to this point, we've been looking at like one gene coding for one phenotype. So, all the examples that I just said. So earlobes and freckles and taco tongue and hitchhiker's thumb, that's one gene for one trait. Most of our phenotypic expressions that have a range or a gradient of phenotypes are not caused by one gene. They're caused by multiple genes, not just multiple alleles, multiple genes. So that was hair color, eye color, skin color, height. Anything that's kind of on a range is going to be caused by many genes. So in this case, they give an example of 3 genes are coded for skin color. And so in this case, they take heterozygous. So heterozygous, you have, you might have HH, EB, FF, just throwing some letters out there. So that would be the white circles and the black circles. So dominant and recessive, dominant and recessive. So you're going to do a tri-hybrid cross, which would be a huge square, which we never get into tri-hybrid crosses. This is kind of what that would look like, 64 square box. And so they're like, okay, so if you make this combination and you get all 6 of your alleles are of the recessive, we'll say that the white circles are the recessive, then your pigmentation might be really pale. If you get all 6 of your alleles to be dominant, you might end up on that end of the spectrum. And then any combination in between, right? So here we have 4 black and 2 white. So wherever we have 4 black and 2 white, we are going to be in that particular range. So this is polygenic inheritance. So you can associate polygenic inheritance with phenotypes that have ranges of expression, not just one or the other, but a range of color or height or things like that. Okay. All right, and then the last environment or the last complex inheritance pattern is the environment. So up to this point, again, we've been talking about things that are found in the genes. Well, we are discovering more and more that we actually have impact on the environment can have an impact on how our genes are expressed. So in the flower pictures, these are hydrangeas, I think. I'm not a green thumb. I cannot grow any plants. Just don't even try. The acidity, if any of you are green thumbs, the acidity of the soil can determine the expression of the flower color. And I don't know what the, acidic or basic, which one's going to give you which color. But if you wanted to manipulate the color of your hydrangeas, you can then alter your soil's pH and kind of push the expression of the flower color more towards the pink or the white or the purple or the blue. So that's in the plant field, but we see those examples in the animal environment as well. So with these Himalayan rabbits, the ambient temperature can affect their fur color in certain locations on their body. So in warm temperatures, they're white all over. When it gets to be cold temperatures, hair cells in their ears, their nose, their tail, and their feet produce a dark fur. So the exposure to cold temperatures changes the phenotypic expression of those particular cells. And one of the reasons why is because when it's cold, the sun, you don't get as much sun rays, and so those body parts that are now black will be able to absorb the heat a little bit better and help them regulate their body temperature. Now there's actually an experiment that people did. This is just a diagram or a drawing of it. They shaved the little bunnies' backs. They put an ice pack on the little bunnies' backs and let that sit for a while, continually, probably not painfully, hopefully not to cause pain, but cold enough to affect these hair cells as they were growing the hair back. They actually got that hair that was white to grow back black because the exposure of the cold. So kind of a cool manipulation of this environment and phenotypic change. All right, so those are our complex patterns of inheritance, kind of a branch off of what Mendel worked with as we are learning more, as we have been able to learn more about inheritance and chromosomes. My last slide is kind of a humorous one. I found this story in this picture, so I asked, what rule of inheritance does this fall under? There's a story behind this cute picture where there was this mama tiger. She was living in a zoo, and we know the reproductive rates of animals that are captive aren't always successful. So she had a litter of her own baby tiger cubs, but they all died for whatever reason. And she was getting really depressed, and they didn't want to lose their mama tiger, right? So they took the little piglets, they wrapped them up in these little tiger print fabric, and the mama just like kind of took them under her wing, and she got happier. She wasn't quite so depressed. Now, I don't know what happened to those little piglets, but I just thought that was a cute way to end this lesson. All right, so thank you so much. That was our chapter 11 material. We'll have a couple more videos from chapter 16 taking a look at specifics in human inheritance patterns and chromosomal abnormalities, and that will wrap up our genetics unit. All right, thank you so much. I will see you next time.